Now that we understand what we mean by ploidy and also what occurs when we talk about chiasmata, let's talk about the mechanics of and the phases of meiosis. And again, just to quickly summarize, when a cell undergoes meiosis, it makes 4 gamete cells which have a ploidy of 1m. The original cell had a ploidy of 2n. And so basically, we start with a 2n cell and it begets 4 1n cells. Now this happens through 2 separate divisions. Each division has 4 phases in each. Phase names are the same in both divisions, but the divisions, which one we're talking about, is noted by Roman numerals. So division And so with 2 divisions, you could guess probably we have division 1 or I and division 2 II. The first division of meiosis, division 1, is referred to as reduction division because this is the division that we go from 2n down to 1n, and so that's where the term reduction comes from. So let's look at all these phases and keep in mind that immediately preceding meiosis we have interphase, during which we have the good old IPMAT, and so these 2 divisions will say we have interphase and we have division 1, which will go through prophase, metaphase, anaphase, and telophase 1. Prophase 1, anaphase 1, telophase 1. And then we have division 2, which immediately follows, in which we have prophase 2, metaphase 2, shift up a little bit, anaphase 2, and then telophase 2. And by the end of this second division, we are going to have our 4 gametes. And once again, they are 1n in ploidy. So in other words, these are going to be 4 sperm cells or 4 egg cells, depending on what type of part of the flower or what type of flower or cone or whatever the case may be this occurs in. So let's go through them. Division 1. We start off with prophase 1, in which the cell pretty much looks just like it did in prophase of mitosis. And let's stick with a cell that has 4 chromosomes. And so here we go. 1, 2, 3, 4. The nuclear membrane dissolves, the chromosomes thicken, become shorter. But one other thing that is, or one thing that sets this apart from mitosis prophase is, write a note here, that the chromosomes align in pairs. And we call these pairs homologous pairs because these are both different versions of chromosome 1 and these are different versions of chromosome 2. What is also important to note during prophase is that crossovers also occur in this phase. And so let me actually change these so that we can actually see some crossovers occurring. Okay, so there's our crossovers, and that's important to note that chiasmata occur during prophase 1. And again, chiasmata are going to dramatically contribute to the variability of the offspring by exchanging different portions of these chromosomes. And then finally, the spindle fibers begin to form, and we can draw a few of them in on our drawing here. And what comes after prophase? Well, metaphase. In this case, metaphase 1. And just like in mitosis, In meiosis, the chromosomes line up at the equator, except this time, instead of a single file line, they line up in 2 rows. Or in this case, columns, the way I have it drawn here. So they become aligned in pairs. The spindle fibers have now fully formed, and they're connected once again at the centromeres, just like they were during mitosis, and we're ready for anaphase 1. So anaphase 1, just like in mitosis, guess what? Well, they get pulled to opposite ends of the cell. But instead of splitting at the centromeres, the entire chromosomes get pulled to opposite ends. And again, this is pulled by the spindle fibers. Let me draw them in. There's still a few going across the middle. And so let me make one note here. The entire, not chromatids, but the entire chromosome are pulled to the poles. So, prophase, metaphase, anaphase. Next is telophase 1, or telophase 1, however you want to say it. And in telophase 1, just like in mitosis, everything gets buttoned up. Chromosomes can uncoil and become a little relaxed. Nuclear membranes form. And we start to see the formation of a cell plate. And eventually we end up with the 2 cells. Each now has been reduced in ploidy from 2N to 1N because we've got 1, 2 chromosomes, 1, 2 chromosomes here. And so the extent to which these chromosomes uncoil and become in an unorganized looking form is species dependent, because in this process, once telophase 1 is completed, most plants are going to immediately go into division 2 of this process. Beyond this is going to be division 2, and that starts with, as you might guess, prophase 2. Okay, so reduction division is completed at this point. All of division 2 basically follows along the same lines, but the only difference is the number of chromosomes have already been cut in half, and we're following 2 cells now instead of just 1. So I'm going to draw my 2 cells. The nuclear membranes dissolve. We're back to just 2 chromosomes in each cell. Okay. Spindle fibers start to form. They line them up. Okay. They connect at the centromeres. And this leads to metaphase 2, which basically looks just like regular old metaphase from mitosis, in that now instead of a double file like we had in metaphase 1, we're only going to see a single file of chromosomes. Let me keep with my color scheme here and write chromosomes in blue. So they line up single file, okay? And this is important to note. So metaphase is where you can really tell what's going on, because if we know that this plant has 4 chromosomes and you saw this metaphase, you could count 4 chromosomes, but if you notice, they're in 2 rows. So we know right off the bat that this would be meiosis and not mitosis. Down here, the only way we can tell these apart from mitosis in this same plant is to count the chromosomes. Instead of 4, we have only 2, even though they're in single file line, and we also happen to be looking at 2 cells simultaneously. So that's another giveaway that this is meiosis. Prophase 2, metaphase 2. Now we're ready for anaphase 2. And things just keep going along their progression as you might expect. We've got two cells. The chromosomes have now split from each other, or the chromatid arms have been pulled to opposite ends of each of these cells by the spindle fibers. And that's pretty much anaphase 2. They migrate to the poles, ana meaning opposite. And that leads us to finish it all up in telophase 2, during which the chromosomes uncoil, they form new nuclear membranes, and we would have already had a cell wall there, but the cell plates begin to form, and by the end they go clear across both of these cells, and now we have a total of 4 cells, each with a ploidy of 1 N apiece, and that is it. And so these cells will either become egg cells if they're found in the female part of the flower, or they'll become sperm cells if they developed in the anthers of the flower. Thanks to the chiasmata, we have not only 4 gametes, but we have 4 gametes which each have their own individual special combination of the genes that are found on their chromosomes. Before we go, let's look at some actual photographs, and I've got a few that we can tell one from the other and what we're looking at. So let's back up here to division 1 again. This particular photo would be from metaphase 1. As you can see, there's a double row of chromosomes here. This is one chromosome lined up with its sister chromosome, or homologous chromosome rather. You can see these are separate chromosomes here. And so this is an example of a cell or a plant that, and these two are too close together to split them apart, that has a total of 2, 4, 6, 8, 10 chromosomes. And this is what metaphase 1 looks like in that particular plant. This photo shows anaphase 1, but you're going to have to kind of take my word for it, in that if you look really closely, you might be able to tell that there are still a pair of chromatids on each one of these groups. They're rather thick to just be single chromatid arms. Basically, this has been turned 90 degrees from this photo that precedes it here. It's in a different plane, but these are the whole chromosomes being pulled in this division 1 of meiosis. So let's continue. This photo shows telophase 1 of division. We can see a cell plate forming here, and we still got in condensed form the chromosomes in either pole. And this figure, or this photo, depicts prophase 2 and division 2. We've already got our 2 cells formed. Each has already been halved in terms of the number of chromosomes, and it's ready to proceed to metaphase 2, which looks like this. These are getting ready to be pulled apart from the centromeres. You can see we've got our 2 cells here. And in anaphase 2, they will be pulled apart. And this photo is probably the best one I've got of division 2, in that you can see in both of these cells, the chromatid arms are being pulled to either end. We've got pulling to this end and this end in both of these cells. And then finally, we have here I would say, telophase 2, in which you can see we've already got one division in this direction. We've got cell plates that are very close to being completed, which is going to result in a total of 4 cells, which would be 1, 2, 3, 4, all of which would be 1N in ploidy, and thereby referred to as gametes.